So, I just got 23andMe today. I'm a little nervous. Trying not to be too excited. Okay. So, I have to wait. Waiting is not my thing. By the way, my college success book arrived today. Because I'm taking a class for getting ready for college. It's not fun. There's a thing on the box about the plan. And I was like, I'm super impatient right now. And I think I'm gonna go the premium because I know nothing since I'm adopted. And Russia doesn't keep good records. And here's Rosie the dog. She is a dog, as you can see, but she acts like a cat. Rosie, show them how you hide behind the cat.
Kit.
Your spit goes on a road trip. Road trip!
The post office scans your kit and we start tracking it in our system, so we know when to expect it at the lab.
That's awkward. I love how these people are talking so naturally about it, but it's what they do every day of their life.
But the journey from spit to results actually began the moment you closed the cap, releasing a special liquid from inside the top of the tube. That liquid gets straight to work breaking down the cells in your saliva and stopping things like bacteria and DNA damaging enzymes. But it's got another trick up its sleeve.
Yeah, so this part's pretty cool. Usually when your DNA double helix is in a low pH environment, also called an acidic environment, like in your mouth, it comes apart.
I have no idea why I'm taking such an interest in this, but I just find this really, really interesting. The science behind this all.
Bonds holding the DNA together are broken.
But the liquid from inside the cap is also working to keep the pH within its happy range.
Critical to stabilizing your DNA until it's ready to be analyzed.
But back to the road trip. Once mailed, your spit might have to travel hundreds, even thousands of miles to reach our lab. Hey Dawn, what's on your spit's playlist?
Where even is the lab anyway?
Genetic jazz? DNA ballet?
No,
Huh.
just the soothing rhythm of the tires moving it closer to the lab.
Well, your spit has no fun. In conclusion, your spit's on its way and we can't wait to welcome it to the lab.
Your spit made it to our lab.
First, we open your kit and scan the barcode on your tube. And a big reason we use those barcodes to protect your personal information and your genetic privacy.
But one of the first things that happens when it reaches our lab is that your sample gets the visual inspection. Does the spit reach the fill line?
Is your spit contaminated with a bunch of other stuff that would mess up the genotyping process?
This is why it's so important to not eat, drink, smoke, or chew gum for 30 minutes before.
I was watching a show where someone was doing a geno process and someone ate a potato chip where they were going to clone potatoes, and the potato turned into a mini hybrid of the person because it was in their mouth. Just imagine that happening.
Spitting.
But if everything's good to go, once your sample passes visual inspection, there's one last step before the real science starts. Remember back when you closed the tube after you spit? The liquid can actually help protect your DNA by keeping it safe in its happy range.
But now there's an even more powerful way to protect your DNA. Scientists use an incubator to heat up your spit sample, which completely stops all the DNA chopping enzymes in your saliva.
And this heating up step doesn't actually hurt the DNA itself, at all. In the right conditions, DNA is actually very stable, and that's one reason why scientists are able to sequence DNA from ancient remains like Neanderthal bones and 40,000-year-old mammoths or Stone Age humans.
That's super fascinating.
After the heating step, we're pretty much ready to go for the next step, extracting your DNA from the cells in your saliva.
See you soon.
Your sample's been taken out of the box, scanned, and heated in an incubator, but your spit's still in this plastic tube.
It turns out that cells from your cheeks and gums
And white blood cells.
make up less than 1% of your saliva, and all that DNA that we're looking for is inside of those cells.
That's awfully complicated.
So what happens now is a pretty amazing process of separating your DNA from the rest of the stuff in your saliva. The first thing we do is something that's a lot like cracking open an egg to get to the yolk.
Except here we're basically using detergents to bust open all the cells in your spit sample, freeing the DNA that's trapped inside.
Detergents? So we're drinking bleach our saliva is drinking bleach.
We do a quick check to make sure there's enough DNA before we go into the next step called amplification.
Which is just a fancy word for copying.
Copying.
This copying phase takes up to 24 hours to make sure that we've got enough copies of your DNA for genotyping.
And then we do something that might seem a little bit counterproductive.
Chop up your DNA.
Your DNA needs to get broken up into fragments, into little pieces, and then we end up with bajillions of little chunks of your DNA in a tube. You still with me?
Yes. Cracking, copying, chopping, done.
Next, we need to clean up your DNA.
We spin your fragmented DNA really fast to separate all the stuff we don't want, like leftover cell junk.
And after this, we're ready for the next step, staging for analysis on the genotyping chip.
So exciting!
So exciting! Your cells.
And today, we get to the headliner of the show,
Genotyping. Genotyping is how we figure out things like your ancestry composition and genetic health risks from your DNA.
How do we do it? We read your DNA like a book.
But we don't actually read every single line of the billions of DNA letters in your genetic code.
Oh. We only need to look at around 600,000 specific spots in your genome, a tiny fraction of all your DNA.
And if you're wondering why we pick those specific variants, it's because they hold particularly interesting information about your ancestry or traits, and they often differ from person to person.
Over the past few decades, scientists have perfected this amazing technology that allows us to read specific spots throughout your genome. It's called genotyping, and it starts with a chip. A genotyping chip.
Unlike a potato chip, this chip is perfectly flat, rectangular, kind of like a stick of gum.
The chip is the key to unlocking your genetic code because it holds a key ingredient, synthetic strings of DNA called probes.
These DNA strips are made of A's, G's, C's, and T's, the same letters your genes are made of.
These DNA strips help us locate the parts of your DNA that are just next to the letters we want to read, called variants.
So when we add your purified, chopped up DNA to your chip,
it's a free-for-all. Millions of tiny chunks of your DNA are swimming around looking for their matching probes. Kind of like trying to find your tiny genetic soulmate in a huge crowd.
aw, that's sweet.
But it can take a long time to find your perfect match in a crowd. Fast forward about 24 hours, and by then most pieces of your DNA have found their probe soulmates.
Once a piece of your DNA hooks up with its probe soulmate, we want to look at the letter in your DNA that comes just after the end of the matching probe.
And to do this, we use colorful fluorescent dyes.
Ooh, color!
We wash your chip with a mixture of fluorescent red and green dyes that are attached to loose DNA letters.
So A's and T's are labeled with red, and the C's and G's are green.
And whatever color letter combo sticks to the end of the probe tells us which variant you have at that location.
That's kind of really cool how they can figure that out and know that. And I feel like this is something most people have heard of from science class. Well, if you were paying attention. This part of science class, I know I was, because I found this surprisingly really interesting.
Well, at this stage, technically it's still just a lot of information about the colors associated with each of the 600,000 plus variants that we care about in your genome. And we decode that color pattern in the next step, analysis.
Yep, and there's one final quality check to make sure that enough of your variants have a color associated with them.
Oh my god, what is it? What is it?
But once it passes this step, we send all that color information to the analysis team, and they'll figure out what all those colors can tell you about your genetics.
Your sample has technically already been genotyped, but it's still just a bunch of color intensities from the genotyping chip that need to be translated into reports that make sense to you.
In the previous step, genotyping, bits of your DNA stuck to matching pieces of synthetic DNA called probes. Through this technology, we were able to take a snapshot of your specific DNA letters or variants that we use to make your personalized reports.
But it's still just a snapshot. Really just like taking a preliminary photo that needs to be color corrected or fully exposed.
But of course, we're talking about your DNA variants, not actual photos, so how does this work?
Scientists on our analysis team receive raw data from the lab and then use computer software to make sense of it.
A super important point here is that your genotype information has been disconnected from your account's personal information, which includes your name, email, address, phone number, etc. Our strict protocols help ensure your privacy and the security of your DNA data.
Keeps things private.
And so this de-identified raw information from the genotyping chip is still just a bunch of colors. The colors carry meaning about hundreds of thousands of your DNA variants.
So for example, let's look at a specific letter which is linked to your ancestry. The color reading from genotyping chip for this variant might be really green. But what does really green really mean?
What the analysis team can do is translate really green into meaningful information. In this case, it might mean that you have 2 copies of a variant associated with Italian ancestry.
You know those commercials where people think they're like 100% Italian and then find out like drastic results? I feel like that's gonna be me. Has anyone else done one of these and found that out, or was it pretty much what you thought? It's gonna be the comment of the day type thingy. I know one of my former teachers who's like my mom. She's basically my mom. And I was asking her questions because she did the same DNA test. And for her, it was what she thought. But what my question would be is, how do they find common ancestors? Also, do they have to take the test to find a common ancestor? Like, I'm sure, my friend who's related to King Louis the 16th, I'm sure he didn't take the test. I'm sure, other people didn't take the test, because how do they have their DNA? How do you know? How do you know for sure? I mean, I understand the close ancestors and how you could know that if they take the test, but far away ancestors, how do they do that? That's a question I don't feel like they answered. Let's watch and see if they answer that.
But if the color was red instead of green, then it would mean you don't have the variant associated with Italian ancestry.
And then there's always the chance that the color is yellow, which meant that you have 1 Italian ancestry variant and 1 non-Italian variant.
Once we have all the correct variant calls, then we use that information to generate your 23andMe report.
Wait. So how do they know it's like, I don't know, Italian ancestry and not like, I don't know, Chinese ancestry? Like, how do they figure out the culture?
And there you have it. Next, you'll hear from us when your results are ready.
I hope you found this as interesting as I found it. And that's basically why I wanted to do a... I'm sorry, I'm overtired. It's like 1 o'clock in the morning and I should be sleeping because I have school in the morning. And I'm not. As you can see, I have bags under my eyes and I don't really care. But I hope you found this interesting because that's why I did this video.